Hi everyone. So my name is Thierra Christopher, and over the summer I did my project called Investigation on Callose Presence in Rhizoids of Ceratopteris richardii gametophytes grown in increasing concentrations of silver nanoparticles. So I did this project along with Faith Smith and Dr. Renee Lopez Swalls, and essentially we looked at how silver nanoparticles, which are in a variety of household appliances like vacuums and washers, burn and wound cream, household disinfectants, basically a variety of things that we use in our everyday life. So we were interested to see how the use of these silver nanoparticles in commercial products, what kind of effect it has on environment, because the more that we have silver nanoparticles in our everyday life, the larger the risk of releases into the environment. And there's kind of an unknown environmental concern currently. So we were more specifically looking at how callose deposition occurs in the rhizoids as the silver nanoparticle concentration increased. So callose is a polymer located in the cell walls of several higher plants, and this deposition of the callose occurs rapidly and in large amounts in response to a lot of stresses like abiotic and mechanical stresses, wounding, pathogen attack, basically anything that could harm the plant. So callose essentially too, it plugs where the cell wall is being attacked. So it basically blocks anything bad from getting into it. So we were using the Ceratopteris richardii or the sea fern to investigate the effects of these silver nanoparticles. More specifically, we were testing their rhizoids, which are thread-like structures which function to keep the gametophyte anchored to the substrate. So we were curious how the exposure of these silver nanoparticles in the growing media, how does it induce callose deposition in the rhizoid cell walls of the sea fern gametophyte? And we believed that if the concentration of these silver nanoparticles increased, the amount callose present in the rhizoids would also increase. So to test this, we started by sowing our spores. We used pre-sterilized spores that were soaked in autoclaved water. We did them under the laminar flow hood. We added 1 drop of spore solution to 2 drops of water, and we added them to our control, or our 0 parts per million, and our treatment, which was 20, 40, 60, 80, and 100 parts per million plates. And then we incubated all of these spores at 27.83 degrees Celsius under constant light and 82% humidity. And then once these spores were pulled at certain days, we did a histochemical staining process 4 days and 9 days after sowing. So we used our germinated spores and we placed them in a 1% aniline blue solution. And we also made a control using buffer solution to test for each treatment group. So then after these spores sat in the solution for 30 minutes to an hour in complete darkness, we viewed the slides under a fluorescence microscope to capture our images. So these here are our images. These were taken 4 days after sowing, and as you can see in the control, there's very little to no presence of callose. And then as you increase in concentration, more callose starts to become present. However, I think you can see it more clearly in our day 9. The 0 parts per million control, there's very little to no callose whatsoever. And then as you increase in concentration of the silver nanoparticles, you can see that there is quite a huge difference between our control and our 100 parts per million. It's very bright once it gets to this point. So we believe that we did find that as the concentration of silver nanoparticles increased, the presence of this callose deposition also increased. And this is because the silver nanoparticle presence in the grain media is an abiotic stress, which causes the plant to use this callose to prevent it from getting in and deposit more into the cell walls. And another thing that I did observe was that when I was pulling the spores from the plates, that as the concentration of silver nanoparticles increased, it was easier to pull the gametophyte out, which tells me that the function of a rhizoid, which is to anchor the gametophyte, was hindered. So for future research, I would like to look into this, and I would like to look into how silver nanoparticles relates to callose deposition throughout the entire phase of the gametophyte and the sporophyte phase, and look at callose deposition in roots. And so finally, I would like to thank the SI Bridges program and Lakshmi. I would also like to thank William Browning for providing me technological guidance, and my colleague David Eltham for providing the 80 parts per million day 9 photograph. Additionally, I would like to thank Dr. Karen Manzaglia for the use of her lab and her equipment to collect my data. And I would like to show appreciation towards the Southern Illinois University of Carbondale for allowing me to use their building and their resources to conduct research. Thank you.